We are 3 students from Mahidol International School, and we are here to present our biology project on the effects of antiretroviral therapy on long non-coding RNA transcription in Thai HIV-1 infected individuals. My name is Pravi Simiruthai, this is Paki Pawatnasuk, and this is Pat Deshav-Schindler. Our project advisors are Ajahn Sathaphon Watanavijan from Mahidol International School and Dr. Bunratasi Draitev from Faculty of Medicine, Siraj Hospital. So, our project is on HIV, or human immunodeficiency virus. It causes HIV infection and develops into the more severe syndrome AIDS. It is currently being treated with antiretroviral therapy, or ART, and it is not currently curable. There's 2 main types of HIV, which is HIV-1 and HIV-2, but the first one is found in most parts of the world, so we're going to be focusing on that. So, the most common question is why HIV is so difficult to cure. That is because of its super complex infection mechanism and viral latency, which acts as a switch from a latent infection to active infection and vice versa, making it invisible from white blood cells. So, our project, what we are doing is that we are examining this thing. It's called non-coding RNA, which is RNA that is not translated into protein. It was ignored in previous research, but now it is found that non-coding RNAs may play a role in establishing viral latency. And current studies on non-coding RNA are mainly focused on HIV subtypes in Europe, so we decided to examine subtypes in Thai HIV patients from the hospital, CRF01AE, and we also decided to examine the effects of antiretroviral therapy, or ART durations as well.
So, our objective of our project, we are focusing on examining the relationship between ART duration and long non-coding RNA expression in HIV-1 subtype CRF01AE, which is commonly found in Thailand. So, our hypothesis, the differential expression margin of long non-coding RNA would decrease proportionally to viral load throughout the duration of ART treatment. So, how we did it? First, we based the experiment on the previous research on HIV-1 subtype B, and we think that what happened in that subtype will happen in our subtype too. So, this is our project workflow on how we do this project. This is our result. This is the graph of patient viral load. So, we collect the samples from each patient from 3 time points. First, before ART. Second, after 4 months of ART. And third, 12 months after ART.
So, looking at the results of our study, you can see that these are the relative gene expression of the selected non-coding RNAs for our study. But since we don't have much time, let's look at this one in particular. This is non-coding RNA ARNC3. This non-coding RNA shows a distinctively higher base gene expression in 2 of the samples, which are sample number 4 and sample number 8. And looking at the right graph, you can see that this is the relative gene expression of this non-coding RNA over 3 time points, which are the baseline, the 4 months, and the 12 months after the duration of the treatment. And you can see that the difference in quantity for each differential expression between the time points, for example in this one, of the 2 samples is relatively higher than other sample. And due to the fact that the baseline of the gene expression is related to the viral load, this corresponds to our hypothesis that the differential expression of non-coding RNA would be related to the viral load, which is directly altered by the effects of ART. And we can conclude that the non-coding RNA expression in our subtype involved with viral load and altered by antiretroviral therapy duration. And this brings us to our conclusion.
Thank you for listening to our presentation. We apologize for the technical error, and thank you.